Welcome to Nanome. Nanome is a platform that allows scientists to collaboratively join spatial computing sessions, look at molecular and chemical structures, modify them, and actually have insights that they wouldn't be able to get using other technologies. My name is Dr. Simon Bennie, and I'm a principal scientist at Nanome. I specialize in quantum mechanical simulations and some molecular dynamics. And today, I'm lucky to be joined by my colleague Chido, who is an expert in this chemical system that he will describe. Chido, would you like to introduce yourself?
Thank you, Simon. My background is in computational chemistry, but rather than doing the high-end stuff that you were doing, Simon, I was actually on the bench. So I've missed a lot of the coding, a lot of the understanding of the dynamics. While I was at LifeArc, I actually worked on a similar system to this. I was working on Mink inhibitors, and we managed to get some amazing insights into how Mink works. Staurosporine is a very interesting molecule. You'll see that it's actually a very big molecule, and if you're looking at it, it fills the whole active site. This molecule is known to hit a lot of kinases. What do I mean? It inhibits, it's almost like a pan-kinase inhibitor. So as far as this system is concerned, it's a fantastic tool compound to show us what happens when this active site is bound to a big molecule. But however, as a compound to follow, it's got a lot of negatives. So today we're going to talk about molecules that can be designed in this pocket that are much more selective than Staurosporine.
Let's take us through this Mink system.
In front of us is a pan inhibitor of kinases called Staurosporine, and it's bound to Mink. So Mink is known to be important for phosphorylating and activating a protein called EI4A, right? And this protein is involved in it's an initiating factor for RNA translation. And now there's a growing list of publications that have associated link with various diseases, including metabolic diseases, melanoma, even diabetes, and other disorders, including dementia. And so Mink has become a very interesting drug target over the years, and a lot of companies have been working on Mink, including where I used to work at LifeArc. Developing some inhibitors of this target might lead us to compounds that could go into clinic to treat some of these diseases. Yep.
So I can obviously see we've got this nice big orange, fairly rigid compound here. I think you said to me before this is called Staurosporine.
So the reason why this compound was used for this particular system is not to prove that it's a drug, but it's been used here as a tool compound, so we could understand, at least the experimentalists who came up with this PDB could understand how Mink binds to an inhibitor. Staurosporine is notorious for binding to many kinases, so it's referred to as a pan-kinase inhibitor. Now let's look at the binding mode of Staurosporine. And using this structure, I'm also going to describe the architecture of this protein, the architecture of the Mink active site, especially the active site of ATP, and also the architecture of the general fold of the kinase. So what we've got here, if you're looking at it carefully, we've got in cyan at the top a subdomain which is in cyan, and then we've got this hinge region here, which is an area between the two, and then at the bottom we've got an alpha helical rich domain. And between these two is where ATP binds. Normally ATP binds with by interacting the adenine core with this hinge region. In this case, what we have is Staurosporine forming a hydrogen bond between the backbone of this thionine here and a hydrogen bond donor pair between. In order for us to actually imagine to look at some of these interactions, we could just in Nanome measure the distance between that oxygen and that nitrogen, and that definitely is within hydrogen bond distance. And you can also measure the distance between this atom and that atom, and as you can see, that is also between in hydrogen bond distance. The angles are correct, and so here is Staurosporine wedged into the cleft between these two subdomains. And at the top, there's this residue here.
This phenylalanine at the back, should we rotate it around so that people can see it on the other side?
We certainly can rotate it around. So what I'm going to do is I'm going to rotate it around so people can see. I'm standing behind it. The reason why it's highlighted here is because in kinase terminology, this is called the gatekeeper. The reason why the people call it the gatekeeper is because this residue seems to be very important in how kinases allow inhibitors to come into this part here, which is called the back pocket. So right now I'm standing in the back pocket. Those people who are interested in designing different types of inhibitors will see that maybe there's an access to the back pocket over the hinge. But more importantly, this aromatic system here is forming a pi-pi interaction between the aromatic system from Staurosporine and the protein, further enhancing the binding affinity of Staurosporine. Other things that you might be interested in.
That's interesting because that phenylalanine is obviously kind of orthogonal to the pi-pi system. So I'm guessing it must have some flexibility to actually generate some pi-pi stacking interactions then.
Yeah. I mean, as it looks here, it will be an edge-face interaction rather than a face-face interaction. Yes, that's correct.
Can I ask if I understood this correctly? Because this is a pan-kinase inhibitor, it's not a good drug candidate because obviously it's non-selective and you want to target specific classes of kinases, if it's a cancer or if it's another type of compound. So what you're going to do is take us through a story about how to take this kind of indicator molecule, this base molecule, and then actually build something up that is being taken to clinic.
That's correct. So in this story, we're going to migrate from how this molecule was used to discover the fold of Mink and have a PDB for it, and how scientists have used fragments to, rather than start with this much more complicated system, but start with much smaller molecules. Today, as we were talking about, we're going to start talking about how we can migrate from the apo structure to the liganded structure. So right now, I'm just showing you these two proteins here, and I'm just going to undisplay one of the proteins. And you can see here, what we have is the position of Staurosporine, and in green here are the positions of the residues that are in the apo structure. So what I'm going to do here is I'm going to show you the structure as it is before it is liganded. And there are some things that you need to see about the structure. The reason why I've highlighted this loop here is because that loop plays a significant part in the way Mink is activated. That's called the activation loop. And the residues that we're highlighting here are the D, F, D. So that's the DFD motif. And in kinases, people will either have a DFG or DFD. In Mink, it is a DFD. And in Mink, it appears, looking at the apo structure and comparing it to where Staurosporine usually binds, which is here, that the DFD loop actually acts as an autoinhibitory loop, i.e., it regulates itself. So it almost auto-inhibits. As you can see here, what it's doing is actually forming an edge-face interaction between phenylalanine 159 and 227.
So that's kind of mimicking what the Staurosporine was doing earlier. Obviously, this edge-face pi-pi interaction. That's interesting.
This is very interesting because you see, if you had this structure alone, without the Staurosporine bound structure, there's very little you'll be able to do to develop inhibitors. But looking at the pocket with Staurosporine, it allows us to actually have enough space in here for us to start developing inhibitors. But this position of the DFD loop is giving us a hint as to how we should be developing those drugs. And if it's a very interesting conformation, actually, because I'm going to just share with us the position of that DFD loop when it's bound to Staurosporine. Yeah, that's the DFD, with the phenylalanine in the pocket, and the loop in the inhibitor bound structure actually starts coming out of the pocket, and indeed is not even resolved because it's that flexible out here. So this is the difference between this part of the protein in the bound structure and in the apo structure. There are many other differences, but this is very key to how Mink operates. And so, everything else is preserved, but the major difference that we can see between the structures is the position of the DFT loop.
Yes, absolutely.
Fantastic. And this has given us a hint as to what else could be done. So since we didn't have a good enough inhibitor in Staurosporine, the story we're going to talk about today is how a research group used fragment screening to find fragments that bind to Mink. And those fragments would be smaller molecular weight, less complicated molecules. And those will be used as the starting point for discovery of a smaller inhibitor, which may be more selective and should exploit some of these conformational differences that we're seeing, in particular in Mink.
And I can see there's a small fragment down here that's very different to what we've been looking at before.
Mm-hmm. Yeah. This is an interesting starting point, right? Because as we were talking before, the people had Staurosporine bound. People didn't really know how to migrate from such a big molecule to something that is drug-like. And fragment drug discovery is particularly useful for finding molecules that are easy for you to develop into drug-like space. And so these are rule of three molecules that are small, that are able to exploit the binding site and lead you to a molecule that might be a little bit more interesting.
There's that phenylalanine. I can see again, you got that edge face interaction with the pi system there.
Exactly. So this small fragment here, which possibly would have been bought from a commercial supplier, is very easy to make, has been co-crystallized, and it was a low affinity inhibitor of Mink. It's been co-crystallized with Mink in order to find a new fold of the protein. Well, what do we mean by that? You can see up here, there's this water molecule here, which interacts with the nitrogen. And this amide is actually sticking in between the DFD loop up here and this lysine here, which is called the catalytic residue, because it usually interacts with the DFT to keep it in position. So in this case, it's actually it's mimicking the phenylalanine in the pocket by auto-inhibiting. Instead of auto-inhibiting, we've got a potential for an inhibitor. You can see when you compare our position where the apo structure, it was putting an aromatic ring here. In this structure, the phenylalanine from the DFT is actually up here in front of me, because it's being kicked out of its own pocket by the fragment. The other key thing to note in this space is that we have potential interactions that are now being formed in this. And I'm going to use my measuring stick again, just to give you a quick understanding of where the distance is between the polar residues. And you can see here, this water molecule here is actually forming some interaction with the lysine, with the fragment, and the D of the DFT loop. And more importantly, we know that there's that edge face interaction or pi-pi interaction between the fragment and phenylalanine 159. But down here, our fragment conserves an interaction with the hinge. Remember, we talked about ATP always interacting with the hinge. But it's interacting here with the residue methionine here, but also interacting with methionines, with the carbonyl from the backbone here of methionine. So you can see, just by observing this little fragment, which is much smaller than Staurosporine, we have mimicked completely the binding pose of Staurosporine. And this gives us an opportunity.
I can obviously see that, you've got a lot more to show me. So I'm guessing the story isn't that they chose this fragment and everything was good. So what's the next stage in this kind of drug discovery process? More fragments?
Well, what you could do, I suppose, and they also did, was look at other fragments. What you're doing with those fragments is trying to map the pocket itself. I.e., you might be looking for fragments that you could grow or bridge between the two fragments to make new molecules, or you might be looking for fragments like this one indeed, which brings in new interactions that you had not seen with the other molecule before. I'm just going to scroll through one or two of the fragments that they did work on, and here are some of the fragments. Let's just undisplay this one, and you can see this other fragment. You can see here, much smaller fragment compared to the other one. It actually has the pi-pi interaction, but there's a water molecule here, and this water molecule interacts with the D from the DFD loop, but actually this fragment has only got one interaction with the hinge.
So there's no water interaction with the ligand here, like in the last compound. So that's interesting.
You don't have that appendage there. So this is a fragment which is exploring the pocket in a slightly different way, actually occupying a little bit further down in the pocket compared to the other fragment, and we can actually align them in Nanome, so you can see the difference between those two fragments as we're speaking about them, and also you can bring in Staurosporine. So you can see the difference between Staurosporine, which is occupying everything, kicking out those water molecules, but in our fragment, which is just making key interactions with the hinge. So you can see Staurosporine is actually being, most of the interactions that Staurosporine is making with the hinge are being mimicked by the fragment. But the fragment is teaching us that you can actually get some affinity without having this big molecule, which is which is quite flat, and people would struggle to make new compounds out of. Here's another fragment.
Okay, so wow, this has got obviously a bit more of a polar system over here compared to the last one, but I can see that conjugated system also doesn't seem to extend as close to the phenylalanine.
Exactly.
The last one as well.
Yeah, and you can see, you could compare those fragments together. That fragment is actually, instead of having that amide group at the top, it's got this lactone group here, right? But it's also interacting with the hinge using just one key interaction. And I'm just going to undisplay the other fragment and then just show you that fragment compared to the last one that we were looking at. And you can see that fragment in pink explores the pocket in a different way. So these fragments and the affinity that they were indicating in the paper, which were low micromolar affinity, showed us that you could actually engage with this site. You could open up the pocket and create new molecules that have a different cause to Staurosporine, but they're much lighter and better in terms of their properties. So, what do you do when you found 3 or 4 fragments bound? Well, the next step is really to start developing molecules, and I'm going to take you from what happened from this fragment to the final drug candidate that was made by this particular group. So the molecule that we're talking about really is already in clinic, and the name of the company is called Effector, and they published this paper in 2019, 2020, and that's when the first molecule, and the molecule that they published is now called in clinic, it's now called Tomivosertib, and Tomivosertib is actually based on one of these fragments, and we're going to explore that fragment. And here the story really does lead into a clinical candidate. We're just going to do a few things. First of all, I'm just going to go quickly to explain why we've got 2 molecules aligned. What we've got aligned in here is Staurosporine in pink and 2 molecules, one in green, one in blue. The reason why we've aligned them in this way is so that you can see the space that is occupied by the new molecules compared to that which is occupied by Staurosporine. Staurosporine has got these multiple rings. these small molecules are much smaller in terms of their properties, in terms of the size. But let me just undisplay Staurosporine so you can see what we're looking at. Before, when we were talking about this, there's this fragment here. This fragment actually sits in the same region as Staurosporine, forming only one hinge region interaction with this nitrogen. You can see where I'm pointing my finger here and sticking this towards where the DFD loop is. And when we compare this molecule with the final compound that this group made, what the experimentalists did when they were making this compound is they put in the cyclohexane group here, and further to that, they put in an amine in this position. And we can just undisplay the fragment so you can see the drug compound bound. And what they've done is, instead of having this pi-pi interaction, H-phase interaction, they've got a methyl group here forming a hydrophobic interaction with the phenylalanine 195. They've got the cyclohexane, which is now occupying the region where the DFD normally sits, and it kicks the loop out of position. And then they've got this lactam ring, if you want to call it, where it occupies the polar region where the activation loop is usually regulated. And more importantly, they've maintained the key interaction with the hinge between this nitrogen and that nitrogen. But looking at this, you would go, all right, why would you end up with a molecule like this? Do you want to see the properties of some of these molecules? And you can actually see why they ended up with this molecule. This molecule is now a preclinical candidate. It's being used in clinic. It's very selective, by the way, for Mink, because it explores the differences in that conformation of the DFD loop. And it actually talks and interacts with the DFD loop while engaging with the hinge, just like Staurosporine does, and keeping the key interaction with the gatekeeper. And you can see we've calculated here the properties of the final candidate. So what we've calculated is in Nanome is the RD kit molecular weight log P, polar surface area, the solubility, you can see here.
Yep.
The number of hydrogen bond donors, acceptors, the number of rotatable bonds, and the number of aromatic rings. You can see Staurosporine has got 5 aromatic rings. And the molecular, and this one has only got 2. Our drug-like compound has only got 2 aromatic rings. Normally what you want to do is to have as low lipophilicity as possible in your drug-like molecule. And this has got a fantastic log P, 1.633, compared to Staurosporine, which is pushing on to 4.5 in terms of our log P.
Obviously, it's a much lighter compound as well for that that log P, sorry. But one thing I can see is that it obviously, according to the AI Cynthia score, the retrosynthetic tool, I can see that, it's slightly less synthesizable, but that, I guess, doesn't matter if you've got a candidate with these properties.
Well, you would hope that it's slightly less synthesizable, because this guy here, which is a CNN, is trained on well-known reaction mechanisms. But these guys would have done a lot of work to come up with this molecule. Because if you go into the structure of the molecule itself, it's got a spirocyclic moiety there, which is very difficult to make. But more importantly, you can see these molecules haven't really gained that much in terms of molecular weight. The fragments that they were dealing with came from about 254, 190, and the molecule that they managed to push into clinic is only around about 300 molecular weight. And actually, it's almost knocking on the rule of 3. This makes it well within the Lipinski rule of 5. Because all these other ones, if you compare Staurosporine, it breaks the rule of 5 in many areas. The molecular weight, the log P, even the solubility is not that great as predicted, and the synthesizability of Staurosporine is not that great. So it doesn't have anything really positive about it. Whereas these fragments here gave you an opportunity to a drug that has got much better properties.
Okay, well, I think that's probably a good place to cap it. Thanks again for all your time, and thank you for everyone joining us in this Nanome session. And we will see you in the next one. Bye, Chido.
Bye-bye, Simon. Thank you.